Hello, and welcome to this video on pioneer transcription factors and the vital role they play in gene expression. In this video, we'll explore how pioneer factors overcome chromatin barriers to activate genes, examine their atomic mechanisms revealed by cutting-edge structural studies, and investigate their expanding roles beyond gene activation, from development and cellular reprogramming to cancer biology. Pioneer transcription factors were first described by Ken Zaret in the 1990s studying hepatocyte differentiation. Unlike canonical transcription factors, which require accessible DNA and cannot engage nucleosome occluder motifs, pioneer factors possess a unique property. They can recognize and bind their target DNA sequences even when that DNA is wrapped around nucleosomes. This nucleosome binding ability comes from specialized DNA binding domain structures. For example, the fork-head domain of FOXA proteins contains a winged helix motif structurally similar to linker histone H1, enabling engagement with nucleosomal DNA. After binding, pioneer factors help open chromatin by recruiting ATP-dependent chromatin remodeling complexes, principally the BAF complex, also known as mammalian Swiss-Neuf, which shift or remove nucleosomes using ATP hydrolysis. They also attract enzymes such as P300CBP and MLL3-4 that add activating histone modifications and sometimes promote DNA demethylation. Together, these changes convert inaccessible chromatin into active enhanced elements, ready for gene transcription. Now, let's look at recent atomic level discoveries about how pioneers interact with nucleosomes. In 2023, Senior and colleagues used cryo-electron microscopy to reveal how the pioneer factor OCT4 binds nucleosomes at atomic resolution. OCT4 uses two DNA binding domains, PAL-S and PAL-HD, to grip nucleosomal DNA near the entry-exit site. When OCT4 binds, it induces a kink in the DNA and partially unwraps about 25 base pairs from the nucleosome. This exposes hidden binding sites OBS2 and OBS3 for more OCT4 molecules and its partner SOX2, enabling cooperative assembly. Remarkably, the N-terminal activation domain of OCT4 contacts the histone H4 tail, rearranging aspartic acid 24 and displacing the tail by over 30 angstroms. This disrupts internucleosomal interactions and promotes chromatin opening. The study also showed that H3K27 acetylation enhances OCT4 cooperativity, inducing a 1 base pair inward DNA shift and exposing more binding sites. While early structural studies focused on single pioneer factors binding single nucleosomes, a groundbreaking 2024 study in Nature revealed something remarkable. Pioneer factors don't just bind individual nucleosomes, they recognize patterns of motifs across entire nucleosome arrays. Using micro-C technology at nucleosome resolution during mouse cellular reprogramming, researchers discovered that OCT4, SOX2, and KLF4 co-localize on arrays of 4 to 12 nucleosomes, spanning 0.8 to 2.8 kilobases. Within these arrays, transcription factor motifs display remarkably specific positional and orientational grammar. OCT4 motifs clustered between superhelical locations 3.5 and 6.5 on nucleosomes, while SOX2 motifs enriched near the dyad and in linker DNA, but critically in orientation-specific patterns. KLF4 motifs flank the arrays at what they termed near and far borders, acting as boundary elements. Feel free to stop the video here to obtain a better understanding of the data presented. The researchers proposed these motif arrangements function as signpost elements that direct combinatorial transcription factor binding to enhancers through a guided search mechanism. In a separate 2024 study, researchers solved the structure of FOXA1 and GATA4, the prototypical liver pioneer factors bound together to a nucleosome. This revealed that FOXA1 binds linker DNA via its fork-head domain and contacts the H2A, H2B acidic patch through its C-terminal intrinsically disordered region. Strikingly, FOXA1 binding caused the nucleosome to reposition, bending linker DNA and exposing a previously occluded internal GATA4 binding site at superhelical location 3.5. This mechanistically explains how FOXA1 pioneers for GATA4 during liver development. Whilst pioneer factors are typically associated with gene activation, a 2024 molecular cell study from Matsui and colleagues revealed an unexpected role in transcriptional repression. During human endoderm differentiation, FOXA recruits PRDM1, a zinc finger transcription factor, which in turn recruits the NURD deacetylase complex and polycomb repressive complexes PRC1 and PRC2. These deposit silencing marks like H3K27ME3 and H2AK119 ubiquitination, as well as the active mark H3K4ME1, creating bivalent enhancers that are accessible but transcriptionally silent, preventing premature activation of genes meant for later developmental stages. This dual role is essential for correct cell fate decisions. Knockdown of PRDM1 led to precocious expression of alternative lineage genes. Pioneer factors are therefore not just door openers, they're master choreographers of gene expression. Finally, what do these discoveries mean for the bigger picture in biology and medicine? Pioneer factor biology has profound implications across development and disease. In development, pioneer factors orchestrate zygotic genome activation. In Drosophila, Zelda activates over 1,000 genes within hours of fertilization. In mammals, DUX4 performs an analogous function at the 2-cell stage. In regenerative medicine, the Yamanaka factors OCT4, SOX2, KLF4, and MYC, 3 of which are pioneer factors, can reprogram differentiated somatic cells to induced pluripotent stem cells, enabling patient-specific cell therapies. In cancer, pioneer factor dysregulation drives oncogenesis. FOXA1 mutations occur in over 10% of prostate cancers, reprogramming the androgen receptor cistrine to drive castration-resistant disease. Additionally, PAX3 FOXO1 fusions generated by chromosomal translocations create neomorphic pioneer factors that establish de novo superenhancers in rhabdomyosarcoma. Over the last several minutes, we've seen how pioneer transcription factors access closed chromatin, drive both gene activation and repression, and shape cell identity across development, reprogramming, and disease. These specialized proteins unlock the genome's potential by directing chromatin architecture and coordinating the dynamic control of gene expression. As research advances, understanding these powerful genome gatekeepers will remain at the forefront of molecular biology. Thank you for listening.